Hi students, last class we discussed about the quantitative analysis, that is volumetric or titrometric experiment we done. Now the qualitative analysis. What is that qualitative analysis? The analytical chemistry deals with the qualitative and the quantitative analysis of the substance. So in qualitative analysis, the given compound is analyzed for the radicals. That is, a simple salt is made up of two parts. One is anion or acidic radical, cation or the basic radical. Acid radical, first you must know the acid radical. The acid radical is a part of an acid and also non-metallic in nature. This qualitative analysis already you done in the class 11. Now the same, only the difference the cations and the anion. Here the acid radical is part of an acid and also non-metallic in nature. It is negatively charged. The examples of the acid radicals: chloride ion, bromide ion, iodide ion, sulfate ion, like this. For example, in hydrochloric acid, HCl, here the chloride ion is the anion. In sulfate, in sulfuric acid, sulfate is an anion. Similarly, the basic radical. The basic radical is a part of a base and metallic in nature, and it is a positively charged. Example, calcium ion. In calcium hydroxide, that is the part of base is what? Calcium. Similarly, in sodium ion, in the part of base of the sodium hydroxide. Similarly, in magnesium sulfate, that is magnesium is the part of the in a substance, the magnesium sulfate. Magnesium is the basic radical. Here a simple salt is formed by the combination of an acid radical and a basic radical. For example, sodium chloride is formed by the combination of acid radical, that is the acid radical chloride ion, and the basic radical sodium ion. So here Na plus and the Cl minus. Each and every radical, whether basic or acidic, has its own characteristics reaction. By doing such reaction systematically, it is possible to identify the radical present in a salt. So such salt analysis is called salt analysis. So many acid and basic radicals are here. We will do few acid and basic radicals. Here you must know that memorize the systematic analysis, that is the general procedure. Then you can do the reaction accordingly. The systematic procedure must be written and the observation and inference must be recorded. Then followed by the reporting of the acid and basic radical analyzed at the end. So this is qualitative analysis. Now we will see the general procedure. Now look at the general procedure. The general procedure is that is systematic analysis of simple salt. Experiment part, observation part, and the inference part. You have to prepare three columns, that is experiment, observation and inference. First, you have to note the color of the substance. In experiment part, color of the substance. The substance shows blue may be presence of copper, pale green may be presence of ferrous, reddish brown means may be presence of ferric, pale pink may be presence of manganese, purple may be presence of cobalt, green may be presence of nickel or Cu2+, that is copper, white or the white crystal, that means absence of the above salt, that means copper 2+, Fe2+, Fe3+, CO2+, Mn2+, Ni2+, like this. Then action of heat. The action of heat, a pinch of salt is heated in a dry test tube. So what does that observation mean? Suppose white turns yellow, may be presence of zinc 2+. Here the maybe you have to use, because this is not a confirmation test. Just maybe, that means that salt may be zinc or whatever it is. So just this is not a confirmation test. This is a preliminary test. Then colorless gas turns lime water milky means maybe presence of carbonate salt. Reddish brown vapors are formed means maybe presence of nitrate. Then flame test. The flame test, a pinch of salt is taken in a watch glass and drop of concentrate HCl is added and make into a paste. The paste is introduced into the non-luminous flame. Bluish green flame means maybe presence of Cu2 plus. Apple green flame may be presence of barium. Brick red flame means maybe presence of calcium. Next ash test. A little amount of salt solution and add concentrate nitric acid and cobalt nitrate solution. A filter paper is dipped in the solution and ignited. The observation is blue color ash may be presence of aluminum, green color ash may be presence of zinc, pink color ash may be presence of magnesium. Then here only we are going to enter the first one, that is acid, identification of acid radicals. First test is action of HCl. The little amount of salt and add some amount of dilute HCl. The water, the observation is water milky, colorless gas with the smell of rotten egg is presence of sulfide. Suppose reddish brown vapors appears means presence of nitrate. Then action of manganese dioxide and sulfuric acid test. Salt is taken and manganese dioxide concentrate H2SO4 is added and heated. The greenish yellow gas is evolved which turns the starch iodide paper is blue, then presence of chloride. Reddish brown vapors, the presence of bromide. Violet vapors evolved means presence of iodide. Then copper turning test. The copper turning test is a salt and add a copper turnings, then add few drops of concentrate H2SO4, the sulfuric acid. The reddish brown vapors means the presence of nitrate. Then the chromyl chloride test, the very important test, the confirmation test for chloride. To small amount of the salt and pinch of potassium dichromate is added and heat. Then the mixture of the concentrate sulfuric acid, that is the that means the red vapors through the sodium hydroxide solution, the red vapors of the chromyl chloride is evolved, so it indicates the chloride is confirmed. Suppose lead acetate is added to the above solution means that is the solution turns yellow, chloride is confirmed. The yellow precipitate soluble in sodium hydroxide also confirmed this about the chloride salt. Ammonium molybdate test. The given salt solution, add concentrate nitric acid and boil. Then add excess of ammonium molybdate solution and heat. The canary yellow precipitate or color is appeared, that formed means phosphate is confirmed. Then magnesia mixture test. The magnesia mixture test, the salt solution or the salt, pinch of salt, then add solid ammonium chloride and boil. Cool, then add magnesium sulfate solution and excess of ammonium hydroxide solution and scratch the sides of the test tube. The white precipitate is formed, that is phosphate is confirmed. Then ester formation test. This is acetate, test for acetate. To the given salt, add ethanol and sulfuric acid, heat, then smell of the test tube. You have very carefully smell the test tube. Already I said how to smell the test tube. So I think you will remember that things. So smelling is very careful. Then the pleasant fruity smell or ester, that is pleasant fruity smell appears means observe means, that is called acetate acetate is confirmed. Then oxalic acid test, amount of substance, there is small amount of substance, then oxalic acid, add 2 to 3 drops of water and rub the paste. That is, again observe the smell, the vinegar like smell means acetate is confirmed. Then the next thing is very important one, preparation of sodium carbonate extract. So they are very helpful to identify the acid radicals. How to prepare the sodium carbonate extract? A small amount of salt, mix twice the amount of sodium carbonate in a 100 ml beaker and add 20 ml of distilled water and boil the solution about few minutes, then cool, then filtrate it. This extract is called sodium carbonate extract. The first silver, after preparation of that sodium carbonate extract, then the extract solution you have to do some experiments, that is identifications of acid radical and also that confirmation test for acid radicals. Here the silver nitrate test. The first take an extract and add dilute nitric acid and few drops of silver nitrate solution. Suppose if a curdy white precipitate is obtained which is soluble in ammonium hydroxide means chloride is confirmed. Suppose a pale yellow precipitate sparingly soluble in ammonium hydroxide means bromide is confirmed. A deep yellow precipitate insoluble in ammonium hydroxide means that indicates iodide is confirmed. Brown ring test. Here they extract and add freshly prepared ferrous sulfate solution and add concentrate sulfuric acid along the side of the test tube. Here very important thing, the ferrous sulfate is freshly prepared ferrous sulfate solution only added. Suppose if you old solution means using of that old solution means already converts to ferric. That's why we have to use the ferrous sulfate, freshly ferrous sulfate prepared solution only. Here the dark brown ring is formed at the junction of two layers that indicates nitrate is confirmed. Then lead acetate test, extract and add lead acetate solution, white precipitate is obtained, the sulfate is confirmed. Then magnesium sulfate test, few drops of extract and add magnesium sulfate solution, the white precipitate is obtained, the carbonate is confirmed, or a small amount of the salt is added and the dilute HCl is taken as the test tube. The colorless odorless gas with a brisk effervescence turning lime water milky evolves. So it's also shows indicates means carbonate. Then calcium chloride test, a few drops of extract with dilute acetic acid, boil off the carbon dioxide gas and add calcium chloride solution, the white precipitate is observed. So that indicates oxalate is confirmed. Then potassium permanganate test, dissolve the substance, that is the white precipitate, in the dilute sulfuric acid and add 1 ml of the potassium permanganate solution, pink color, that is pink in color, the discharge with this steady effervescence, that is indicate oxalate is confirmed. The copper turning test, substance taken in a dry test tube and add some copper turnings and add concentrate sulfuric acid, no characteristic changes, so absence of nitrate. Chromyl chloride test, this is barium chloride, so chloride confirmation test. The amount of substance, add a pinch of potassium dichromate and heat with concentrate sulfuric acid, pass the red vapors through sodium hydroxide solution. So red vapors of chromyl chloride is confirmed about the chloride and the solution turns yellow, the chloride is confirmed. Again lead acetate is added on the solution, yellow precipitate is obtained, that yellow precipitate soluble in sodium hydroxide, chloride is confirmed. So the confirmation test for chloride. Then preparation of the sodium carbonate extract, prepare the sodium carbonate extract and extract with the silver nitrate test. Silver nitrate test means add the silver nitrate test in the extract. the curdy white precipitate is obtained that is soluble in ammonium hydroxide, so chloride is confirmed. Then identification of basic radical, preparation of the original solution. Already I said how to prepare the original solution. Some amount of substance, add water, that is called original solution. After confirmation of chloride, so you have to do the directly basic radicals, that is group separation, identifications of basic radicals, the group separation, also called group separation. First group, that is for a 0 group, 1 ml of the original solution and add Nessler reagent, and excess of sodium hydroxide solution is added. No characteristics precipitate is obtained, so absence of 0 group. Then do a first group radical, original solution with dilute HCl, no characteristics precipitate, so absence of first group radicals. Then few drops of original solution with dilute HCl and then pass hydrogen sulfide gas, no characteristics precipitate is obtained, so absence of lead copper As3 plus. Then few drops of original solution, add ammonium chloride and ammonium hydroxide solution, no characteristics precipitate, so absence of group 3 radicals. Then to few drops of original solution, 1 ml of ammonium chloride and ammonium hydroxide solution is added and pass H2S gas, no characteristics precipitate, so absence of zinc, cobalt, nickel and manganese, so that is absence of group 4 radicals. Then confirmation test for basic radical, because barium, I gave the barium chloride, so group 5 is barium is present, so that potassium chromate test, few drops of original solution with potassium chromate, yellow precipitate is indicate, barium is confirmed. Dilute sulfuric acid test, original solution with dilute sulfuric acid, so white precipitate is obtained, which is insoluble in HCl, mean barium is confirmed. Okay, so this is the systematic way to write the procedure. So after confirmation test both acid and base, you have to report the salt. Here barium chloride, barium is the basic radical and chloride is the acidic radical. Students, we will perform this salts. Usually in the examination we don't tell what is the salt is given, you have to identify with the help of the systematic procedure. Okay, here I will show you, we are going to do this salt in this year: magnesium sulfate, ammonium chloride, copper sulfate, aluminum sulfate, ammonium carbonate and lead chloride. These 6 salts we will do the confirmation test here, but you have to do the systematic procedure. I will show you the confirmation test for these substance. Now we will do the experiment, students. Before that I will show you the apparatus for the salt analysis. This is watch glass, this is test tube holder, tongue, then glass rod, dropper, beaker, funnel. This is test tube stand, test tubes and boiling tubes, wire gas, the tripod, stand, then the Bunsen burner. Now we will do the experiment. See, the next salt is ammonium carbonate. Already we did ammonium chloride, this is ammonium carbonate. Test for, now I am going to test the acid radical. Here acid radical is what? Carbonate. So take the substance and add dilute HCl the brisk effervescence comes. So it's indicate the brisk effervescence. So this indicates carbonaceous presence. Suppose sodium carbonate extract test is there, means yes, there is also one test, that is magnesium sulfate test. Already I prepared the sodium carbonate extract. Take the extract, small amount of extract, then add magnesium sulfate solution. The white precipitate is obtained, means, can you see the white precipitate? So this shows the white precipitate. So carbonate is conformed. The acidic radical carbonate is conformed. Now we will do for basic radical the test. We will perform the test for basic radical. Prepare a original solution. Take a small amount of the substance and prepare the original solution. With the help of glass rod, dissolve the substance. And now we will do the test. Take a small amount of the substance. Then add Nessler's reagent. The brown precipitate is obtained. So here this indicates what? Then addition of the sodium hydroxide solution is added. Red is brown precipitate or brown precipitate. So this is reddish brown precipitate. This indicates ammonium is conformed. Now next test is sodium hydroxide test. This is the original solution. I am going to add sodium hydroxide solution and warm So the characteristic smell is evolved, that is the ammonical smell. So the smell is evolved ammonical smell, so ammonium is confirmed. Now you can see the test for acid and basic radical. So this is basic test for basic radicals. Ammonium is confirmed. See, look, confirmation test for basic radical and the confirmation test for acidic radical. The acid is what here? Carbonate, and basic is ammonium. Ammonium is brown color, precipitate is obtained, and white precipitate for carbonate, that is the magnesium sulfate test. Okay, now we will do the next salt.